नमस्ते, हमेशा खुश रहो बिटिया. I am JVN Dr. Lily Trivedi and today I am going to take the lecture of Genetics Lab, the preparation of slide of Chironomous larva for observing polytene chromosomes. Now let's start. What are polytene chromosomes? Now, the salivary glands of diphtheриan insect larvae have nuclei which are always in the interface. Now, they were described by Balbini in 1881. The nuclei of these chromosomes have abnormally large size as compared to the chromosomes of the other body cells. Now, because of their large size, they are also called as giant chromosomes. The giant chromosome, if fully extended, are 100 times longer than those normally found in metaphase. The DNA of the giant chromosome repeatedly replicates itself, but the daughter strands do not separate. So the replication is nearly 100 times, resulting in 1024 parallel strands held together. A fully developed banded chromosome are nearly 25 to 0.55 nanometer long. The chromosomes also show alternating dark and light bands, and each band represents a gene. The amplification of the DNA of salivary glands of chromosome in the insect larva is known as polytenization, and these chromosomes are referred to as polytene chromosomes. So now let's discuss the procedure. Now what is the procedure for Now we'll discuss the procedure. Now what is the procedure for um, yeah, let me share with you. Now when we have the preparation of salivary glands is already done, the sample taken is here is Chironomous larva. Another name of Chironomous larva is bloodworm. Now, now Balbini first reported polytene chromosome in these larvae. Now, Balbini rings are exceptionally large puffs on the polytene chromosome in the diphtheриan Chironomous larva salivary gland. So, what are the materials required? First, we require a fourth instar Drosophila larvae, or it can be Chironomous larvae. Two pointed needles are required. Microscope, slide, cover slip, dissecting microscope, acetoalcohol 1:1 fixative, acetoarsine stain, 45% acetic acid, a compound microscope, a large watch glass or petri dish, blotting papers, and 1N HCl. Now we dissect the Chironomous larva. We require the help of pointed needle. With one needle, we will hold the posterior end of the larva and pull the anterior part with the second needle. Then we locate the salivary glands and remove the other debris. After this comes the stage of fixation and staining of salivary glands of Chironomous larva. We fix the salivary glands with acetoalcohol fixative for 5 minutes. Then remove the excessive fixative with the blotting paper and add 2 drops of 1N HCl over the salivary glands to hydrolyze them. Then the slide is incubated for 2 minutes at 57 degrees Celsius. Excessive HCl is removed with the blotting paper. 2 to 3 drops of acetoarsine stain is added over the salivary glands of the larvae. Then we cover the slide with a petri dish or large watch glass and wait for 5 minutes for the stain to take. After 10 minutes, for 10 minutes only, and after 10 minutes, the excessive stain is washed with 45% acetic acid. And then we take a blotting paper to remove any excess of stain or acetic acid. Then we add 1 drop of acetoarsine stain over the Chironomous larva and cover it with cover slip. Now, after we have made the slide, another procedure is there, that is squash preparation of this glands. Now, the salivary gland under the cover slip, we place the slide between two folds of blotting paper and apply pressure with the thumb over the cover slip and roll it. Now, after we open the blotting paper, then we seal the edges of the cover slip with DPX or nail polish. Now, what are we going to observe? The observation when observed under that microscope is polytene chromosome. Now, as we make sketch, we will see that this portion is the polytene chromosome. This is how it is going to appear in the microscope. And this picture, both the pictures I have taken from Wikipedia. So this is all about the experiment of how we are going to prepare the slide. So students, I hope you have liked my lecture. If you have any comment or query, please give in the comment section. I will resolve it soon. This session was powered by digital version 2.0 of Jyoti Vidyapeeth Women's University. Thank you and namaste.